Welcome everyone to the Society for Developmental Biology first annual postdoctoral seminar series. We are very excited today to highlight the work of our outstanding postdoctoral members. And today we have Camila Lopez-Anido from Stanford University and Fanju Meng from University of Rochester, who will share their amazing research. Each speaker will be given 20 minutes to talk, followed by 10 minutes of Q&A. To the audience, please enter your questions in the Zoom Q&A box. You can do so throughout the talks or at the very end of the talk. Today, our first speaker is Camila Lopez-Anido. She did her graduate work in the lab of John Svarin at the University of Wisconsin-Madison, where she investigated gene regulatory networks that define myelinating glial cell identity in mammals. For her postdoc, she joined the lab of Dominique Bergman at Stanford, where she pursued her interest in understanding how gene regulatory networks guide the remarkable plasticity of cell fate decisions in plants. And her work is supported by the NIH F32 postdoctoral fellowship. Today, she'll tell us about her work in elucidating cell fate decisions that underlie developmental lineages in plants. Camila, please go ahead.
Thank you for that introduction. I am excited to be part of this seminar series. As a molecular geneticist and developmental biologist, I'm really interested in these flexible lineage dynamics that can arise when you're in the process of building a tissue, such as this leaf here. And I'm particularly interested and drawn to plants because they display extraordinary biological strategies as sessile organisms, responding to different environmental cues to enable the appropriate physiological responses and developmental responses as well, of course. And plants have taught us that there are many ways to be successful and overcome challenging environments. So we can take advantage of unique features from plants to explore how underlying flexible lineages are controlled. This helps shed light not only in foundational principles that in developmental biology, but also helps provide insight into how plants may respond to changing climates. To get at this with our model organism plant Arabidopsis thaliana, for which there are a range of molecular genetics tools available, we leverage the accessible epidermal tissue, which you can see here in this cartoon. Mature tissue is comprised of two cell types. We have in purple here 2 cell stomata that form a pore to enable the exchange of gases with the atmosphere. This is essential for photosynthesis and transpiration. Stomata are surrounded by pavement cells that form this protective epidermal barrier for the more specialized tissue underneath. And we can have fewer or more stomata, essentially changing the cell composition of the epidermis depending on environmental cues. These can be light temperature, humidity, water availability, or changes in carbon dioxide levels. So the lineage that's giving rise to these two different outcomes here exemplifies adaptable, flexible cell fate decisions that we can use to learn more about how cells are coordinating to build an epidermis that's going to ultimately match the given needs of the plant. Like a lot of lineages, this one begins with a series of asymmetric divisions. With a protodermal cell on the left here, it will divide asymmetrically to yield a larger cell and a smaller one. The larger cell is known as a lineage ground cell, and the smaller one is referred to as the meristomoid. Either of these can go on to divide asymmetrically once, twice, 3, or 4 times. And this can play out in a few different ways, but I'm just showing here 2 outcomes. On the top, we have the division of a meristomoid cell dividing asymmetrically, and on the bottom, the larger lineage ground cell dividing. This is notable because each of these stem cells will at some point commit to different cell fates. So how these divisions play out is quite important. Meristomoids commit to becoming guard mother cells, which will divide symmetrically now once and yield a pair of guard cells. These will undergo terminal differentiation and cell expansion to form this pore. The lineage ground cells in white ultimately undergo endoreduplication, and they'll differentiate into pavement cells. So how does this actually play out? How are these processes coordinated in a living tissue? In this one frame of a time lapse here, we can already see cell outlines on the epidermis of this growing, developing Arabidopsis leaf. We have nuclear markers here that are indicating where early lineage cells and blue mid-lineage cells, the meristomoids and the guard mother cells are. You can see they're dispersed on the leaf. And I want you to note that because these cells are all constricted by their cell walls and they're limited to one plane on the epidermis, we can actually track asynchronous cell divisions and also fate decisions. So let's zoom in to one section here. You'll notice that there are cells of different shapes and sizes, and we have different nuclear markers expressed here. You may have also noticed these two cells in the center forming a young stomata, but I'm going to ask that you focus on this meristomoid here. So in the time lapse, this meristomoid will divide symmetrically, and the cell further from the stomata will then commit to the next stage in the lineage, this guard mother cell fate. Now, this time lapse is going to repeat here, but what I want you to think about is how there's some kind of order enforced into cell fates, because presumably in the next frame of this time lapse, this mid-lineage cell will divide symmetrically to yield a guard cell that's spaced apart from the one that's already there. Our nuclear reporters are encoding BHLH transcriptional regulators, speechless and mute. These are respectively required for initiation of the lineage and then commitment to guard cell fate. Now, note that speechless is expressed prior to every asymmetric division in these multipotent stem cells, but its expression is only maintained in meristomoids and is then followed by expression of mute as cells transition to the guard mother cell fate. Our lab and many others have shown that environmental cues can modulate signaling cascades that eventually converge onto speechless, and this is a way to fine-tune asymmetric divisions, which ultimately are impacting the final cell composition of the leaf. These are followed by FAMA. This is a third BHLH transcription factor in the lineage, and it would be expressed here in these young guard cells, but I want you to note that it is required for terminal differentiation to bookend the lineage. So we do tend to think of these as these three factors as working in combination with more broadly BHLHs. These are regulating sequential stages in the lineage. And interestingly, this does parallel lineage functions that are ascribed to other BHLHs in animal systems, such as in muscle development. In our system, what is really remaining unclear is how these cells are transitioning through these identities. So in other words, how can we reconcile this model of discrete cell stages, knowing that cells are influenced by their neighbors in a living tissue, as well as by the broader environmental context of the plant? So we hypothesized that there may be some heterogeneous cell states, particularly within the early flexible stage shown here, but we would also perhaps gain better resolution into transitions like to the guard mother cell fate, and then as through differentiation, as these cells are committing to final cell fate, stomatal cell fate. And having insight into these transitions would enable us to gain new perspective on foundational questions. So we can come back to thinking about how are asymmetric divisions controlled, but also what is establishing fate commitment and what determines terminal identity. So when I joined the lab as a postdoc a few years ago, I sought out to define these cell fate decisions. And I was really excited to use single cell RNA sequencing in combination with molecular genetics in Arabidopsis. This would let us gain insight into potential processes that we wouldn't otherwise be able to uncover, but also we could take advantage of the amendable genome of this small model organism plant. So today I'm gonna share what we've done to reconstruct some trajectories and also identify gene modules in our system. In our approach, we've isolated stomatal lineage cells using two reporters. One is shown here on top, and it's selectively expressed in our lineage. The other reporter enriches for epidermal cells while also capturing inner tissue. So we could make comparisons between other lineages in the plant. So starting with leaf tissue, we isolated cells by protoplasting to break down the cell wall, and we use fluorescence activated cell sorting to capture cells for single cell RNA sequencing. We use SmartSeq2 and 10x genomics technologies to capture hundreds or thousands of cells at different read depths. And I'm first going to share what we've done to gain perspective on the lineage by comparing it with others in the developing leaf, and then we'll take a closer look at dynamics within the lineage. So focusing on the developing leaf first, we use standard pipelines with Seurat version 3 to get a map of cell identities, represented here by this UMAP dimensionality reduction, where every dot is a cell and cells are grouped based on similar gene expression. so we can visualize relationships between cells. And I want you to note that the axes represent many dimensions from all the cells. So we're going to simply use this notation here on the bottom left, and we're going to focus on the spatial distribution of cells in relation to each other. We used graph-based clustering to identify potential cell states, shown here with different colors, and through differential expression analyses, with also reference to well-characterized genes from the literature, a few of which are shown here in these violin plots of expression across different clusters, we were able to assign cell identities that account for the main tissues of the developing leaf in the upper corner. You can see a cartoon of a cross-section where we have epidermis encasing the inner tissues. So two main larger clusters of epidermal cells were found, and we were also able to detect mesophyll, so the primary site of photosynthesis, as well as vasculature tissue, which is required to transport nutrients and water throughout the plant. To build on our framework, we asked whether we could infer lineage dynamics using latent variables that may be hidden in the data. So what I mean is there anything else in our data that we can capture, essentially? So we use RNA velocity and SE-velo to layer on mRNA splicing dynamics. They're represented here by arrows that are on top of the UMAP. These are predicting future outcomes of cells based on the ratio of nascent, unspliced transcript and mature spliced mRNA transcripts. By reconstructing these lineages within different tissues of the leaf, we could then identify tissue-specific modules, comparing them to each other. Of course, with a focus to better understand the genetic programs in the submodel lineage specifically. And so to briefly summarize one module that we focused on and followed up with Arabidopsis genetics, we extended our understanding for how mutant FAMA coordinate cell cycle progression through the G1S, G2M phases. And we found that cyclin D7 works with common regulators such as the kinase WEE1 at this critical step. So to take a closer look at the epidermis and to further refine cell fate decisions in our lineage, we profiled more cells, which were enriched by the lineage-specific reporter. We examined the relationship between cell states, shown here in this UMAP, with a more stringent program called Slingshot to predict pseudotime. And this is represented by the color progression and the black line overlaid on the UMAP. Pseudotime reconstructs potential lineage transitions or dynamics. And with graph-based clustering, even though these are relatively arbitrary cutoffs, we're also able to see a gradient of gene expression profiles in these violin plots when we align clusters through pseudotime on the x-axis. Based on these profiles, we were really excited to see a continuum of early stem cell identities, which are shown here on the left. This is what we more or less predicted we might see, but we weren't sure whether they would be closer together or really quite discrete. So with this continuum, we can really think a little bit more about how formative early lineage decisions may be impacting cell fates. So given the single cell resolution here, we could also infer onset of differentiation. Essentially thinking based on gene expression, these cells up here are on their way to becoming pavement cells, which weren't captured in the data because we were enriching for early lineage in our approach. We were also able to capture the major stages in stomatal differentiation, where guard mother cells divide symmetrically and differentiate into guard cells. And with this framework, we've asked, of course, whether we could identify new players in lineage transitions. So we selected a few candidates to follow up with that are differentially expressed across pseudotime, and we think maybe coordinating with known regulators. So I'll just share one example with this gene here. This gene encodes for a transcriptional regulator SHINE2. It's been implicated in cuticular wax structure and in stomatal patterning, but it otherwise hasn't been looked at very carefully. It appears to be expressed in guard mother cells in our UMAP here, which is colored by the cluster annotation. So we asked if whether we could also see this in vivo using reporters. With a transgenic line expressing YFP NLS under the SHINE2 promoter, we assess different stages of lineage progression, which is shown here from left to right. And we found that expression is absent from early lineage cells, but is enriched in guard mother cells and in recently divided guard cells. We no longer see expression in mature stomata. The yellow in the center here is autofluorescence from the stomatal pore opening. So we're now trying to understand what are the functional roles of SHINE2 and other candidates, and how may these be coordinating lineage transitions? We've defined here a new view of our lineage comprised of these heterogeneous cell states, which enables us to ask questions and kind of notice things that we wouldn't otherwise be able to notice. And we're following up to identify and test mechanisms that control these flexible fates downstream of environmental cues. But we also found a surprising twist in the biology, specifically when we were thinking about how continuous cell states could be regulated by our discretely acting factors. This question really got to us. So we thought that we might see some overlap, as can be observed in other systems, but that's not what we saw. So let's take a look at where these factors are expressed in our single cell resolution data sets and consider new perspectives that arose. So in our UMAP here, you can see that mute is expressed tightly in guard mother cells. And so the color is indicating cluster annotation. So these cells here have detectable mute expression. Mute precedes expression of FAMA, which comes on in this late GMC cluster and is expressed in guard cells. When we look at cells that express both mute and FAMA, we thought we would see somewhat of an overlap, but we only found 3 cells that express both of these. So the domains really are quite discrete, and we're still thinking through why that is and how that could be controlled. This is kind of the first, we should really think more about how this transition is happening that we otherwise may not have appreciated. On the other hand, speechless expression was found broadly throughout the early lineage stem cells, which you can see up here. As expected, we thought that speechless may be playing different roles in all of these stem cell identities, and it may have different functions. We'll have to follow up with that. But you may have also noticed that speechless is expressed in guard mother cells. This was a bit of a surprise, but we can also find a portion of speechless cells that are expressing mute and a portion that are expressing FAMA. This is something we weren't looking for, but given this finding, we revisited the reporters that we had, and we saw speechless protein expressed in guard mother cells and also in new guard cells in vivo. So we next asked, wait, is speechless playing a role to regulate differentiation? Have we overlooked something? And so before I share what we found, I want to remind you that we really had no previous indication that speechless may regulate these late stages, because genetic approaches really place speechless function earlier. I'll show you what I mean. So speechless is required for lineage initiation. When we compare wild type control here on the left with the speechless null on the right, you'll notice that while we see stomata pseudo-colored in purple at a given time point in wild type, they're largely depleted from the epidermis of the speechless null on the right. And these plants are also quite sick. Turns out stomata are very important. To test the role of speechless later in the lineage, we used a stage-specific knockdown with artificial microRNAs driven by the mute promoter. And we found that a reduction of speechless transcripts within guard mother cells led to a reduction in stomatal counts. So you can see a representative DIC image here, but also quantification on the right. What got us really excited is that we also found this diverted cell identity phenotype, pseudo-colored in yellow here. And this is representing a new phenotype we haven't quite seen before. So we were really thinking about what might be going on. If you look at the division planes outlined in white, it appears that cells have undergone spiraling asymmetric divisions in a few rounds. So we might have had this first larger cell, and it could have divided through the middle here, let's say. And then perhaps we could have had another division here, another one here, and then here. So you can kind of see how the cells were spiraling through their divisions. And at some point, they kind of forgot who they are and they failed to generate stomata. We would normally have a stomata there in the center. We quantified this phenotype also, again, in 2 different lines. And we thought a little bit more about how speechless might be enforcing differentiation. And to get at this, we're returning to our single cell resolution data sets to identify candidates that, for example, may be expressed at this given critical window where speechless appears to be required to seal the fate of differentiating guard cells.
1 minute.
So to recap the major takeaways, we've identified tissue-specific modules in the developing leaf, leveraging insight from different tissue layers that's made accessible through single-cell RNA sequencing. We've also defined this continuum of stomatal lineage stem cell identities represented here in our schematic that we're following up with and really excited about. And through single-cell RNA sequencing and molecular genetics, we've been able to appreciate that speechless plays a broadened role to enforce late cell fate decisions and to yield stomata. Our work is available through bioRxiv, and we're currently going through revision, so I welcome feedback on that. And moving forward, I'm really excited to use this framework and this system to think more about how cell fates are controlled, and also how underlying genetics may define climate change adaptation and evolution. And so with that, I would really like to thank my postdoctoral mentor, Dominique Bergman here, and her group, and particularly the people that contributed to the project that are bolded, and especially for the work that a previous postdoc, Ani Vatten, did to re-examine speechless expression in vivo. I'm, of course, grateful for the communities and resources here at Stanford and for funding sources, especially for the NIH, for supporting my postdoctoral fellowship. And I welcome questions. Thank you.
Thank you, Camila, for that beautiful talk. I should mention that Camila is on the job market this year. So a reminder to our audience to please type your questions directly into the Q&A box in Zoom. And while we wait, I have a question of my own. I noticed that in the big cluster of stem cells, the speechless expressions seem to be quite heterogeneous. So some cells had it, some cells did not. Can you comment on that a little bit more? And what does that indicate?
Yeah. Yeah, so thank you for that question. What you're seeing here is you notice, I think what you're noticing is that there may be more speechless expression in this bottom half of this larger cluster as opposed to the top. And what that is are meristemoids that, remember, the ones that will retain speechless expression prior to mute coming on. So it is really nice. We were expecting to see some speechless expression in these lineage ground cells as well, because they need to upregulate speechless before they divide. But it was nice to see kind of a gradient of speechless expression also within our lineage.
Great, thank you. We have a question from Zainab Afzal. Really neat work, Camila. I was wondering what would happen if the time for cell division was increased or decreased. Would that have any change in cell fate, since spatial resolution seems very important here?
Cell division. Yeah. So I'm wondering if the question is about the timing of cell division, whether cell division. Yeah, I'm not sure that I understand the question. Could you repeat it, maybe?
Yeah. She was wondering what would happen if the time for cell division was either increased or decreased, and would that have any impact on cell fate, since spatial resolution seems important?
Yeah, so cell division timing is important. And there's one idea that asymmetric divisions and symmetric divisions are utilizing different factors and are really quite different processes. So we don't have concrete evidence for that yet, but that is a question that we're interested in thinking more about. So I think that's all I can say about that for now.
Thank you. So next we have Michael Piacentino. He says, beautiful results. Does mute directly bind to the FAMA locus, or do you have any candidates for transcriptional regulation that act downstream of mute to control the timing of FAMA expression?
Yeah, so that's kind of a question still as well. We know that FAMA will bind and repress a lot of other players and processes, but I'm not sure whether mu is turning on FAMA. I'd need to check that. Yeah.
Sounds good. If we don't have any further questions, maybe we will turn it over to Duan to introduce the next speaker.
Thank you.
Thank you.
Okay, good afternoon. Our next speaker is Fanju Meng. He's from the University of Rochester, and he performed his graduate training with Benoit Patou. Excuse me, sorry. Benoit Patou, who's investigating the requirements for SOX transcription factors in the adult stem cells that maintain and repair Drosophila intestines. Now he's in the Murphy lab at the University of Rochester, where he's using zebrafish to investigate the epigenetic control of neuroquest differentiation. And today he will share his research titled Dynamic H2AZ Patterning Controls Chromatin Accessibility and Neuroquest Differentiation Trajectory. Thank you.
Thank you, Dequan, for the introduction. I'm very excited for this opportunity to be here to share my ongoing work in the lab. I'm very interested in the transcriptional control of cell-fate transitions. Cell-fate transitions are controlled by these cell-fate specific gene expression programs. And here represented by these different lines that these represent a cluster of genes that are unique to each of the cell-fate. This specificity is in part generated by this differential accessibility of the genome to the each of the cell-fate. And this epigenetic states define these unique chromatin configurations that controls this differential accessibility. And epigenetic states is unique to each of the cell-fate, and it helps to serve as a barrier to maintain the cell-fate. And this is not fixed. Changes to the epigenetic states also regulate either lower or increase the barrier, and this regulates the dynamics of cell-fate transitions. Epigenetic states can be regulated at multiple levels. For example, regulation of chromatin accessibility is one aspect. And here, there's some regions in the genome are restricted, while there are regions in the genome are more accessible. At the nucleosome level, where H2AZ is wrapped around, post-translational modification of the histone tails and the replacement by the histone variants play important roles in regulating epigenetic states. We are particularly interested in the role of H2AZ. H2AZ is the histone variant that's highly conserved during evolution. It's found nearly in all eukaryotes. Compared with the ubiquitous H2A localization, H2AZ is enriched at gene regulatory elements, for example, gene promoters and enhancers. It's involved in this gene transcription and DNA regulation, replication. We are trying to address these two open questions. So how H2AZ genomic patterning regulates epigenetic states, and how H2AZ patterning impacts gene expression and cell-fate transition in vivo. To address this question, we use both mouse embryonic fibroblasts method and zebrafish embryos. Today, I will first share a study of how H2AZ regulates chromatin accessibility. This is a collaborative study with Kristen in the lab. So we first look at published dataset of H2AZ localization and chromatin accessibility by ataxic. And here in this heat map, we rank all the genes based on H2AZ level. So genes with high H2AZ are on the top. We then plotted chromatin accessibility data by ataxic. in the same order. So here you can see that genes with high H2Z also are more accessible. In addition, H2Z localization also correlates with gene transcription level from the RNA-seq. So this pattern holds true in both mouse embryonic stem cells and the MEF cell. These results show that H2Z resides at active genes and highly accessible chromatin regions. So does H2Z regulates chromatin accessibility? To address this question, we turned into this AMP32E mutant. AMP32E is the H2Z chaperone that antagonizes H2Z incorporation. It has been shown by others and us that in the AMP32E mutant, there's increased H2Z level in both MEF and zebrafish embryos. So in this way, AMP32E is a nice tool to test the effect of H2Z changes on chromatin accessibility. So we then performed a taxic to check chromatin accessibility in this AMP32E mutant. And here is a log2 fold change of AMP32E mutant chromatin accessibility to the Y-type ataxic. Here, shown by this yellow color, there's a widespread increase of chromatin accessibility in the AMP32E mutant. As quantified in this differential attack size, there's 95% with this size will have increased chromatin accessibility. So we hypothesize that this is due to the H2Z expansion in this mutant that leads to this increase in chromatin accessibility. So if our hypothesis is correct, then knocking down H2Z should restore the changes of chromatin accessibility. So we knock down H2Z in the AMP32E mutant and then performed ataxic. As shown here, this is a log2 fold change that's comparing the H2Z knockdown in the AMP32E mutant to the AMP32E mutant itself. And as shown by this blue color, chromatin accessibility is reduced. These results show that H2Z knockdown rescued the chromatin accessibility change by AMP32E loss. So far, I showed you that data that H2Z patterning at the gene promoter regulates chromatin accessibility. I don't have time to go over all other results. We have shown that H2Z patterning at the transcription start site also regulates transcription factor binding and the gene expression program. This study just came out 2 months ago, so check it out if you are interested. So the big question is, what's the function of H2Z patterning during cell-phase transitions in vivo? Would a similar mechanism that we found in MEF hold true? To test this question, I turned into zebrafish embryos, which is a really powerful in vivo system to study developmental cell-phase transitions. We have the AMP32E mutant fish in the lab. Interestingly, AMP32E now embryos shows variable phenotype across development. You will see some lethality across different developmental stage. One interesting phenotype I want to emphasize that in the AMP32E now fish, they appears to have larger jaw compared with the Y-type. This phenotype is quite exciting to me because the jaw structure in the craniofacial skeleton are derived from neocrest cells. Neocrest cells are a specific population of cells that are unique to vertebrates. They are derived from the neuroectoderm, but they have the striking multipotency during development. They will generate not only ectoderm-derived tissues, they also generate, for example, connective tissue and cartilage that are mesenchyme-derived tissues. In zebrafish, neocrest cell-phase transitions, their neocrests are specified around 10-hour post-fertilization at the end of gastrulation. After their induction and specification, they will start migrating across the body and then differentiate into multiple cell types, including the cartilage in the craniofacial skeleton. So we have the H2Z chipseq and the gene expression profile from this AMP30 null embryos at the 6-hour post-fertilization. So we first check the relationship between H2Z localization and the gene expression. As I mentioned earlier, in the AMP30 mutant, there's H2Z expansion across the genome. And here, if we look at this specific locus, FOXC1A, we saw this increase in H2Z level. Interestingly, when we check gene expression level, we saw this induction of FOXC1A at this in the AMP30 mutant. This pattern also is found for PAX3A and many other genes. So these results show that in the AMP30 mutant, H2Z expansion leads to gene activation. We are very interested in the nucleus cells based on the phenotype of jaw in the AMP30 mutant. So we are particularly interested in to check nucleus gene expression signature. This study recently identified gene expression enriched in the nucleus cells using salted cells. And here we performed this gene set enrichment analysis to compare our gene expression program in the AMP30 embryos to those nucleus genes. Now we found that nucleus genes are enriched in this gene expression program in this AMP30 mutant. I want to emphasize that this gene expression profile are from 6-hour post-fertilization. And you expect to see nucleus gene signature until later around 10-hour post-fertilization. So this is our working model. Normally, nucleus cells are specified around 10-hour post-fertilization, but in the AMP30 mutant, where we saw H2Z expansion, even at the 6-hour post-fertilization, we saw some signature of nucleus gene expression. Currently, we don't really know the exact mechanism how these genes are activated. We hypothesize that this is similar to MAF cells, where we show that H2Z expansion leads to increased chromatin accessibility and transcription binding. So we hypothesize that in this AMP30 mutant, this H2Z accumulation leads to open chromatin and increased transcription binding, and that results to this nucleus gene signature. I'm testing this hypothesis by looking at chromatin accessibility and the transcription binding in this AMP30 mutant. As I mentioned earlier, AMP30 antagonizes H2Z incorporation. Another key player that's really important for H2Z patterning called SR-cap. SR-cap is responsible for deposit H2Z in the genome. When I check the SR-cap mutant fish, the homozygous mutant fish shows smaller jaw size compared with the control, and it also has this heart edema during development. And these defects are consistent with the role of nucleus cells. This result suggesting that SR-cap is important for nucleus cell development. So I followed up the jaw defects by performing iosin blue staining to look at the developing cartilage. Here is what Y-type cartilage looks like. And here in the SR-cap mutant, if you focus on this middle area, this specific cartilage called serrato here is flattened. as quantified by the angle between these two cervitocele. In addition, there's a stronger defects in this posterior cartilage called cervitobranchial. And in the SRCAP mutant, most of the cervitobranchial are missing. So based on the Elsing blue staining, it seems that the anterior cartilage are not affected in the SRCAP mutant, while there's a stronger defects in the posterior cartilage. This phenotype is quite interesting because the anterior posterior patterning phenotype is also inherited by this anterior posterior patterning of the nuclear cells. And here, nuclear cells that are migrating to the anterior pharyngeal arch will generate the anterior cartilage, while nuclear cells that migrating to the posterior pharyngeal arch will generate the posterior cartilage. So based on the phenotype of SRCAP mutant, I have hypothesized that this may be due to defects in nuclear cells. To identify these nuclear cell defects, so we performed in situ hybridization of the nuclear cell specification markers FOXD3 and TFAP2A. Now here in the SRCAP mutant, we didn't detect this change in the expression pattern, and these results suggesting that nuclear cell specification probably not affected in the SRCAP mutant. These results indicate that this craniofacial cartilage defects might be due to defects in later stage, like migration and differentiation at the pharyngeal arch. So we recently got RNA-seq data at 36 hours with SRCAP morphant. And here, when we look at expression of this gene DLX2A, which is a marker for nuclear cells that migrated to the pharyngeal arch, we saw this slightly reduction in the DLX2A expression. So currently, I'm testing this hypothesis that this craniofacial cartilage defects are due to the nuclear cell defects at the posterior pharyngeal arch by looking at more nuclear cell specific markers. One classical factor that distinguishes the anterior arch with the posterior arch is the Hox genes. And the Hox genes are very well known to be important regulators of the anterior posterior patterning. When I look at the RNA-seq data in the SRCAP morphant, for example, this Hox A2B, which is expressed in the anterior arch and seems not affected, while Hox D11A, which is expressed in the posterior arch, have reduced expression. So currently, I'm also testing whether the Hox gene changes in the Hox gene expression might contribute to this anterior posterior patterning defects of the craniofacial cartilage. So here, opposite to AMP32E, where we saw H2Z expansion, and we saw this nuclear cell gene signature prematurely activated. In the SRCAP mutant, we saw we have H2Z reduction, and we found defects in the craniofacial cartilage. And these defects, these anterior posterior pattern defects, might be due to nuclear cell migration or differentiation defects. So our central hypothesis is that H2Z reprogramming occurs during these nuclear cell phase transitions. So currently, I'm using zebrafish genetics and genomics to really identify the changes of H2Z localization accompany each of the cell phase transitions, and then compare that to chromatin accessibility and gene expression profile to identify these reprogramming events that facilitate nuclear cell fate transitions. So with that, I want to thank everyone in the lab, especially Kristen for the collaborative study of math, and also Patrick and Claire for their help with zebrafish work and data analysis. Our collaborator Travis Spiller at SUNY Geneseo, our founding resource, and the Society for Developmental Biology for these wonderful opportunities. I'll be happy to take any questions. Thank you for your attention.
Thank you, Fanju, for that very nice talk. You have several questions. So the first question is, does M32E and SR-RAP affect H2AZ, only affect H2AZ? Are there any other functions of these proteins?
For the AMP32E?
Both for AMP32 and CR-RAP.
Okay, yeah. So not much knowing for the AMP32E. So it was identified as H2AZ chaperone. So right now the major function for AMP32E is to inhibit H2AZ installation. So really not much knowing about other function in terms other factors. And for the SR cap is known to install H2AZ. So both of the functions are really tightly to H2AZ localization.
Okay, so the next question is from Ondine Cleaver. Nice work. Your CR-RAP mutants appear to have cardiovascular defects. Did you look at NeuroCrest's contribution to heart development?
Yeah, that's a great question. I haven't followed up with that defects. So yes, in the SR-cap mutant, they have this heart edema, and they might have this vasculature and circulation defects, but we haven't followed up on these defects. It will be interesting in the future to check whether NeuroCrest cell are the lineage, for example, in the heart, and are also affected or not.
So the another question from Christopher Wright is a Boletti et al. 2020 paper published in vivo evidence that the absence of mitosis, that in the absence of mitosis, H2AZ is not needed for transcription. Thus, H2AZ on active promoters is a marker, but not an active driver of transcription. What is this? What do you think about this related to your working model?
Regarding the H2AZ, so that's controversy about H2AZ role in transcription. What I think is that H2AZ as the promoter will set up the stage for transcription. So H2AZ at the location will increase chromatin accessibility, and that will increase transcription factor binding. So H2AZ has already shown to be facilitate the transcription factor binding, for example, FOXA2 and OCT4 in stem cells.
Okay, thank you. The next question is from Nabora. Hi, you saw an early activation of NeuroCrest genes at 6 hours. Did you lose expression of other genes important for the embryo?
That's a great question. I haven't really checked in like gene expression for other cell fate. So I can't really tell from there, but we saw this enrichment for this NeuroCrest gene expression. One ongoing work is to using single cell technology to really at the population level to look for this gene expression signature and see whether this H2AZ expansion might bias some of the cell fate while at the expense of other cell fate. That's a great question.
The next question is from Brandon Carpenter. The next two questions, I'll ask them together. Maybe the first question is, what does expression of major signaling components like hedgehog or notch genes look like in your embryonic expression analysis? And the second question is, what does neuronal patterning look like?
Yeah, for the first question, so in the AMP32 mutant with the disrupted gene expression, we definitely saw several factors in the signaling pathways, for example, Wnt signaling and Hox genes, and also BMP signaling pathways. But I haven't really tied to the specific embryonic phenotype at this particular time points. Can you repeat the second question?
Yes, the second question was, what does neural patterning look like in your mutants?
Yeah, in this AMP32 mutant, I haven't done much to look at the neural crest gene expression. So we are currently using neural crest cell markers and reporters to look at neural crest cell in this AMP32 mutant.
Okay. I think the question was about like neural, like the
okay.
Yeah.
Yeah, so that one we haven't followed up. So I don't know that one.
The next question is from Ed. In wild-type organisms, H2AZ is enriched at promoters and enhancers. In the mutants, the AMP32E mutants, is spread aberrantly. Does it mean that AMP32 is targeted to aberrant sites to remove misincorporated H2AZ? What targets AMP32E?
Yes, that's also a great question. So H2AZ in the AMP32 mutant H2AZ expansion, that happens at both promoters and also enhancers. So we think that H2AZ expansion in this promoter and enhancer to regulate their function. But how AMP32 is targeted to the promoter where how AMP32 specifically to inhibit H2AZ at some of the promoters, that has not been well studied.
Okay, the next question is, great story. Did you look at the role of H2AZ? Do you think the role of H2AZ will be found for other cell type transitions as well, or is it something specific to NeuroCrest? And if so, why?
Yes, great question. So I will start with the SR-cap phenotype. So in the SR-cap mutant, we saw H2AZ reduction, but the phenotype so far, it seems there's stronger defects in the craniofacial cartilage. But otherwise, the animal, there's some defects in heart edema, but otherwise the animal looks just not much difference with wild type. So it's possible that a subset of cells, for example, NeuroCrest cells, are particularly sensitive to H2AZ manipulation. And that might be due to the special of NeuroCrest cells that have this multipotency and really diverse differentiation potential. That might, because during this process, there's H2AZ reprogramming event. And that's what I'm currently working to test this hypothesis.
Okay, you have 2 more questions and you have time, so we're gonna keep going. Have you looked in any NeuroCrest transgenics like SOX9 to see if the migration of the differentiation or the differentiation of NeuroCrest cells is affected?
Yeah, so in the SR-cap mutant, I haven't done in situ to look at SOX9, but based on the gene expression profile, I haven't saw clear reduction of SOX9 expression. But it could be due to migration defects at this later stage for the NeuroCrest cells. Currently, I'm using more markers to look at migration and the later differentiation at this SR-cap mutant.
Okay, and I think you effectively answered April's question, which is, will you look at neurocrest cells in the SOX reporters like the SOX10 GFP? And so sounds like the answer is yes.
Not based on the reporter. We haven't done that. But for the gene expression, it's not much affected in the SRCAP mutant.
So I guess the question is, are you going to evaluate your mutants with these different transgenic markers like SOX10 GFP?
Yeah, so we have SOX10 GFP and the SNEL1B GFP. So I'm currently checking their reporter expression across development in the SRCAP mutant. But so far, I haven't got the results for that part.
Okay. That was our last question of the session. So thank you, Camila and Fanju, for really great talks. The seminar will be recorded and will be available on Monday on the website. So check out there. In the next seminar on January 8th, we will hear from two speakers, Christopher Bolt from the Akil Polytechnique Federal de Luzon and Nicole Edwards from Cincinnati Children's Hospital Medical Center. Thank you and have a good time.